Thank you very much for coming to this session. My words are cautiously optimistic. So we've heard a lot this morning about optimism, and hopefully we'll be able to continue that as we move forward for the next hour. My name is Tim Cripe. I'm a physician scientist at Nationwide Children's Hospital, affiliated with the Ohio State University in Columbus, Ohio. And it's my privilege today to be among a distinguished panel here and moderating this morning's session on gene therapy. We have a distinguished group, including Dr. Jerry Mendell from Nationwide Children's Hospital, Jorge Quiroz from the CMO from Solid BioSciences, Tara McDonnell Moorhead, the Director of Clinical Research from Pfizer, and Kevin Flanigan from Nationwide Children's Hospital. And we're going in a surprise order this morning, and to start us off is Dr. Flanigan.
Good morning. Thank you. Thank you for the opportunity to talk about this project, which is surrogate gene therapy with RH74 MCK GALGT2 for Duchenne muscular dystrophy, and just present some very preliminary results and a safety profile from the trial. A few disclosures, but are not really relevant to this study. So the idea of surrogate gene therapy is based on not replacing dystrophin, but putting in a gene that can substitute for function of dystrophin. This is a cartoon of the dystrophin-associated glycoprotein complex, which you're all familiar with, that shows dystrophin binding actin at one end and binding beta-dystroglycan at the other end. And via that connection through the sarcolemmal membrane to alpha-dystroglycan, and alpha-dystroglycan binding to myrosin or laminin alpha-2. And that binding is dependent on the glycosylation status of dystroglycan. So dystroglycan glycosylation is critical toward the function of the muscle. But in fact, there's two such complexes. Although we always show the one with dystrophin, there's a second dystroglycan complex that's located at the synapse, at the neuromuscular junction, and also at the myotendinous junction. It differs from the dystrophin-associated one across the extrasynaptic membrane because of the presence of utrophin as the binding partner, a paralog of dystrophin as a binding partner, and also because of the presence of a GalNAc transferase, an enzyme that is responsible for a particular mode of glycosylation of dystroglycan there. So this is encoded by the gene GALGT2. As first shown by Paul Martin many years ago in dystrophin-deficient mice, as in boys with Duchenne dystrophy, the dystrophin is absent at the extrasynaptic membrane, although the synaptic complex is intact and utrophin is there and functions properly. It turns out when you overexpress GALGT2, as first shown by Dr. Martin in 2002 in transgenic animals, GALGT2 overexpression results in expression of GALGT2 across the synaptic or the extrasynaptic portion, the entire muscle membrane, and restoration of utrophin across that entire membrane as well. So this was, in fact, it's important to know that this is not solely utrophin dependent in transgenic animals and also in non-human primate studies. Overexpression of GALGT2 results in expression of multiple membrane-associated proteins, including agrin, alpha-7 integrin, plectin, several of which have been shown independently in animal models to show overexpression partially corrects the Duchenne, the MDX phenotype. So it's not simply a utrophin-dependent mechanism. Dr. Martin showed that in transgenic animals, there was a lack of muscle damage when you cross these transgenic overexpressing animals with MDX. MDX is here marked by the squares. This is a measure of serum creatine kinase activity in those mice. And you can see that throughout the life of the mouse, there's an elevation of serum CK, just as we see in boys. The GALGT2 activity results in a particular epitope called the CT antigen. So you'll see in these slides mention of the CT antigen. CT antigen just is a marker of activity of GALGT2. And in the transgenic animals that express it, you see that there is no elevation of serum creatine kinase through 6 months in the animal. Correlating with this is the absence of muscle pathology. This is the same model. MDX mice marked by the squares. You can see that there's significant muscle pathology as we measure it over the life of a mouse by internal or centralized nucleation, a marker of muscle degeneration and regeneration. So in contrast, the CT antigen presenting animals, the GALGT2 animals, have essentially really no muscle pathology for nearly the entire lifespan of the mouse, as marked by the black squares. So quite prominent protection. This is true, and not just in transgenic animals, but also in AAV treated animals. Here is the AAV construct that's being used in the study with MCK promoter and GALGT2. And in the top left in panel A, there's you can see force drop. This is a measure that we use in mouse models of muscular dystrophy with repeated contraction of the muscle. There is a drop in the amount of force. There's a normal drop in force. This happens with the black line there. You can see wild-type mice. In the MDX mice, this force drop is accentuated. And in GALGT2, the green line is hidden by the red line because they really overlap. They show similar degrees, microdystrophin and GALGT2, similar degrees of protection against force drop. In fact, a little supraphysiologic correction. And similarly, specific force is improved from the MDX mouse. It's indistinguishable from wild type and indistinguishable from microdystrophin. And these values occur with levels of about 20% of muscle fibers expressing GALGT2 in the mouse model. So we're optimistic, enthusiastic about GALGT2 because it shows broad therapeutic potential in the muscular dystrophies. In fact, I'm sorry, I skipped one there. There seems to be an animation that didn't make it over. Not only the DMD model of MDX, but the LGMD 2D model, alpha-sarcoglycan deficient mice, and a model of congenital muscular dystrophy, a mouse called the DYW, congenital muscular dystrophy type 1A, or Merison deficient congenital muscular dystrophy. Animal models of each of these show improvement when treated with this. Second reason we're enthusiastic is because this is not a new gene being put into the body. This is an endogenous gene. We all already express GALGT2, so it's turning up a gene that's already there. So we don't expect issues with transgene autoimmunity in the same way. And finally, this would be agnostic as to what mutation or mutation class there is in DMD patients. It would be independent of what the mutation was. So we've initiated a trial under IND 16175. This is a trial that is currently using intravascular limb infusion for gene transfer. This is delivered via the femoral artery to the lower limbs. The femoral vein and artery on each side are cannulated, and I'll show you some images of this in a moment, and catheter localization confirmed via angiography. The blood flow is interrupted with inflation of a balloon, both on the venous and on the arterial side, and then the vector is given over 90 seconds and remains in the circulation for a 10-minute dwell time. After the 10 minutes, Dr. Martin has shown that this shows an increased uptake of muscles in the leg. After this, then the venous and arterial balloons are deflated. And in fact, the vector does go into the general circulation, and it's not removed. But this is not dosed nor intended as a systemic delivery in this study. It's a local regional delivery. Now, using this in non-human primates, this is a new paper in Press from Dr. Martin. This is ILI delivery, as we call it, to non-human primates allowed us to establish the dose escalation, the proper dose. And you can see two different doses here, the vastus medialis muscle from two different non-human primates. And the one on your right shows the dose we're using as the first level of dose in the human study. That shows, these are pictures from different sections of this sort of cartoon representation of a muscle, that throughout the vastus medialis muscle, there's quite significant numbers of fibers that express GALGT2, ranging from about 22% to above 80% of fibers, all within a range that we would expect a physiologic benefit based on the mouse studies. This is planned as a dose escalation phase 1/2 trial with a low dose at 2e to the 13th vector genomes per kilogram per leg, or for a patient, 5e to the 13th vector genomes. And then a high dose that was double that amount, 5e to the 13th vector genomes per kilogram per leg, or 1e to the 14th. The primary objective of this is safety. And the primary efficacy outcome is expression of the CT antigen by immunofluorescent analysis to show the same degree. This study is not powered, obviously, to show a clear, although we're studying efficacy outcomes, including a 100-meter walk time and the North Star ambulatory assessment, it's not powered. The primary outcome is actually on expression, immunofluorescent expression. So we've enrolled one patient at present who was enrolled at nearly 8.5 years old with a duplication of exons 22 to 41. And when he enrolled, he was on a relatively low dose of prednisone at 10 milligrams per day. We treat from the day before injection through a period of about a month and a half afterwards on prednisone. You can see at enrollment, he could walk about 357 meters on the 6-minute walk, about 60% of the predicted value for a boy his age. Similarly, on the 100-meter walk time, he was about at the 60th percentile compared to boys his age as well. His North Star ambulatory assessment was 18. And then we measure in just the muscles that were targeted, muscle flexion and extension. We can measure force in kilograms as well. This is an MRI showing a gradient echo image through his legs prior to initiation of therapy. And we've highlighted the vastus medialis muscle because that's the muscle that was biopsied at 0 and 4 months in his case. And you can see, for example, the vastus lateralis and other muscles that show fatty replacement by a high signal. This is to show you, you can see the two balloons inflated here, the dark sort of sausage-like images there, one in the femoral vein, one in the femoral artery. And then confirming by angiography the distribution of this throughout the whole leg, directly into the leg. This is the right femoral artery. And we monitored the legs during this, and he had no, rather, except for a little bit of modeling of the skin, had no detrimental effect of this. So there were the adverse events of bruising at the catheterization site, a completely expected adverse event, resolved by day 30, and a decrease in the absolute lymphocyte count that we've seen. We made changes to his steroids as well at the same time. None of these were unexpected. His serum transaminases, his AST and ALT, peaked, as we've seen on multiple patients do, about 14 days after we did the gene transfer. And there's a decline of about 43% from his screening serum CK, continuing his usual activity at home. And we saw nothing except a transient elevation of what's called an ELISpot test, looking for T-cell autoimmunity generated by the therapy. Some of the data, this is a very brief look at the data compared to baseline to day 120. Some measures, in the top one, the measures we show here as a percent change from baseline. The 6-minute walk test declined slightly by about just over 10%. This is actually just outside the range of what we consider retest variability. But there are improvements in 10-meter walk, improvements in 100-meter walk time, and improvements in strength and force measurements in kilograms in 3 out of the 4 muscles tested. Certainly this isn't enough to show that there's a clear functional improvement, but there's no convincing evidence of decline suggesting that the procedure itself was safe, and we'll look for the remainder of studies over the coming months and year. This is very preliminary. It's an unblinded analysis of the CT antigen. So this is not our final. Our final analysis will be done in a blinded fashion with a validated assay that's under development. But one can see, as opposed to the pretreatment one in the top and the no primary control, no antibody in the bottom, there is actually widespread expression of the CT antigen, certainly above the level of 20% that we set as a target. So we can say that this is well tolerated, no convincing evidence for a functional decline at 4 months post-injection. We have had a couple of patients who've failed screening based on serology and one other for a medical reason, but in fact, we anticipate now under moving with our DSMB to go directly to doing a second patient at the higher intra-arterial dose, having shown proof of concept for expression of the vector and safety of the procedure. And then Dr. Martin has just completed dose-finding studies that will support systemic delivery, and in fact our plans are to jump, after a second patient at the higher dose, to jump directly to an intravenous systemic trial. So I'll close by highlighting that this work was funded by actually several groups, including Team Joseph and Little Hercules Foundation. But that the trial is funded by the NIH under an R21, and acknowledging Paul Martin and whose work it really is, as well as a large effort from many people in our institution. So thank you very much. We're doing it.
Thank you, Kevin. We've decided we'll do all 4 presentations before questions and answers. That way we can go for Q&A all at once. So next up is Dr. Jorge Quiroz from Solid BioSciences.
Good morning. Thank you so much for the invitation and for the opportunity to let you know about Solid BioSciences, what we're doing, why we are here. We have, during this presentation, some forward-looking statements. We're a public company since January. I'll tell you a little bit about that. So we have to make the disclosure. And perhaps to spend 1 minute or 2 to tell you who we are. Solid BioSciences was created with the objective of cure DMD, which is a very ambitious objective. We are a company entirely focused on Duchenne muscular dystrophy. We're not searching for other neuromuscular disorders. And the reason it's quite obvious. The company was founded 4 or 5 years ago by a family with a patient with Duchenne and their friends and their families. That was Annie and Ilan Ganot. Annie is here today. And with a lot of help with other patient associations, particularly in the UK, Kerry Rosenfeld, and Alex Johnson and Emily Watson, that are now part of the different charities, Duchenne UK and Duchenne Research Fund, also here today, were very pivotal on getting this moving forward. By then, the idea was to cure Duchenne. It was very agnostic to which kind of treatment that could be or that could like. And in fact, a panel of experts was put together and decided that although very ambitious, gene therapy had the highest chances of being transformative on the field. And that ended up being hopefully true, or we hope that it will end up being hopefully true. And now we have a couple of companies working on the same field, for all of which make us very, very, very happy. We are focusing on all the patients with Duchenne, every single mutation, any phenotypic characteristics, despite of the stage of the disease. That is our target. It's very ambitious, but that's what we are, and that's the reason why the company was funded, and we're trying to follow suit to that commitment. For the reasons that I have mentioned, we're very closely attached to the community, patients with DMD. Since early on, we're interacting with PPMD and other associations in the US to get their input on how to move forward. And now in January this year, we made a big step from a private company. We became a public company in order to get funds to what was coming. And now we're more than 80 employees. We have a new headquarters in Cambridge. We have our own labs. We have a lab for research and development. We have a lab for process development, which means what you need to do to manufacture the gene therapy or gene transfer, which is not an easy task. And we have been working very heavily on that for the last 3 years. We have also called to help us the group of leading experts on gene therapy and in Duchenne in particular. We have an experienced scientific advisory board. I see here Stan Nelson and Caro Micheli. This is their son, who have been helping us on every aspect along the way in the last 3 years, with many others, Dr. Barry Byrne at the University of Florida, that himself have experience on AAV9, on manufacturing processes, on dosing patients. We have been working, and I'll tell you a little bit about that, with Dr. Chamberlain's big set of microdystrophin construct, Dongsheng that I see here as well. So trying to do the right thing, what we put together a very thorough package in front of the FDA to open an IND, which is the kind of documentation that allows you to start studies in humans. On that, we had a very extensive program with animal models of the disease, with animals that didn't have the disease. They're trying to explore the right dose, and with that, we went into the clinic. As a big overarching way of understanding what is gene therapy, as you see here, it is made up of particularly 3 elements, slightly more than that, but in synthesis, 3 elements. One is the gene. The gene, as you know, is the gene for the dystrophy protein, and I'll tell you a word about that in the next slide. The second is the vector or the capsid or the virus on which the gene is incorporated. And the third aspect is the promoter. The promoter is a section of the gene that goes right before the gene that encodes the protein that allows that protein to be expressed in the most efficient way in different tissues. So when you combine those 3, what we achieved is to be able to administer this gene in a way that is systemic, so it's through the veins, to try to target all the muscles in the body. The muscles that are the skeletal muscles, the diaphragm for the respiration, and the heart. And for that, we selected a capsid or a vector that is called AAV9. We have a promoter that is called CK8 that is specific for muscle. This combination targets the heart, the diaphragm, and the skeletal muscles in a very impressive fashion. From what we were able to observe during our preclinical studies. And perhaps a critical piece is the gene. So the dystrophin gene, as you know, is a very large gene, is the largest gene in the human genome. And what we have done, and this is based on the work of Dr. Chamberlain and Dongsheng to identify the functional components of the gene. Not all the pieces of gene were created equal, and through an engineered design, not trial and error, engineered design, we're testing which portion of the genes were the important ones, and with that, they were able to create a gene that was small enough to fit inside of that viral particle that I showed you in the slide before. So all of these, plus the promoter, needs to be in a small amount, but still needs to be functional, and not only functional, but of course to able to express the protein. And that is what we packaged together. We call the microdystrophin 5, and with that, we are able to produce the new form of the protein. We're calling it the microdystrophin protein, which is the one that will accomplish the function of the naturally occurring dystrophin that the patients with Duchenne, of course, do not have. And with that, I will tell you that what we're doing with the gene therapy program. We were able to file a package with the FDA last year, and they allow us to move forward to patients. We thought a lot about how to conduct this trial, and this is the design that we came across. This is a randomized study, which means that patients are coming and they are by chances being allocated on the treatment arm of the study or in a control group. It's an open label randomization, so although chances play a role in who goes to the treatment versus who goes to the control group, once that randomization happens, the patients do know in which group they are. So as of now, we have 2 patients. I'll tell you more about that. One in the control group that is untreated. There is no placebo here. The advantage of doing it this way is that that patient, the one in the control, doesn't need to go or undergo biopsies, and you will see that in a second, versus the treatment control. This is the word control here, so we can compare outcomes between these 2 groups. It's open label because the patients know to the group that they are located, and it's a single ascending dose design. In general, the first time that you go in humans with one treatment, you want to understand the dose efficacy relationship. So you start at a dose that you think that is efficacious, and then from there you go to the next dose that could be 2 times or 3 times that dose, and then again 2 times or 3 times that dose, and you're ascending that dose. It's a single ascending dose because gene therapy is administered only once, but even for drugs that are administered multiple times, the way to start with a single administration. What we're planning to do is to enroll up to 32 patients, 16 in the control arm and 16 in the treatment arm. And we are trying to target all patients. So we are enrolling patients that are ambulatory, the children, and patients that are non-ambulatory, the adolescent patients. And we have started the trial at the University of Florida. The objective of this trial is, of course, safety. We want to understand if this construct, these three pieces together that are being administered systemically through the veins to patients, it is safe. But at the same time, we want to understand if it is efficacious, and efficacy here defined in a very discrete manner. We want to understand if the gene is producing microdystrophin. And that will be the first step. Of course, we're also searching on all the functional outcomes that you are very familiar with, all the tests that you are familiar with. Because we have a proportion of patients that are adolescents in this trial, we're also searching for respiratory function, cardiovascular function, to understand if a transformative amount of microdystrophin will express in a transformative functional improvement. And this is the way that the trial looks like. This is an artificial representation, but it's the best way that we could put it together. There is a screening phase for the patients to get into the trial. It may take 1 or 2 months. The patients, as we discussed, are randomized into the treatment arm, which is the orange on the top, or the control arm, which is the gray at the bottom. For the patients who are randomized to the control arm, those who are not going to receive treatment immediately, after 1 year, they're going to give the opportunity to receive the drug, if they will, and they are still able to receive the treatment. So that's called a switch on the treatment arm. And on the control arm, we have different cohorts. The cohorts are the different doses, and here you have the way that it looks today. Those letters in between the orange arrows, DSMB, means Data Safety Monitoring Board. So before ascending to the next dose cohort, we're asking a group of experts that are independent from us, from the company, and they're expert in gene therapy, an expert in Duchenne. We group them together, and one statistician. They're going to see if the data collected is safe enough for us to continue moving forward. We propose the way that to move forward, and they will accept or reject that proposal. In that way, we're preserving the safety of the patients that are participating in the trial. The last thing I would say about the design is that the patient in this trial are going to undergo 3 biopsies. This is something that we also thought a lot. And the biopsies are going to be 1 at the baseline before treatment, and 1 at the end of the treatment of this first observable period, which is 12 months. We want to understand, though, how the microdystrophin, how long does it take for the microdystrophin to express over that year? So we have asked the patient to give us the opportunity to look at 3rd biopsy, which we call the intermediate biopsy, in between these 2 time points. And for that 3rd biopsy, we have different alternatives on when to obtain that biopsy, at 1.5 months, 3 months, 6 months, 9 months. We want to understand how it goes. So every patient will have only 3 biopsies, which we understand is a lot, but no more than 3 baseline endpoint at 1 and the intermediate time point. And the inclusion criteria are here for you to know. They're also all available in clinicaltrials.gov. The patients, they need to have a clinical diagnosis of DMD and documented dystrophin gene mutation that is predictive for DMD in the case of the children. As I was mentioned earlier during the morning, some of the patients may have antibodies against the viral capsid that we utilize to infuse the microdystrophin gene into the patients. And that proportion varies. These viruses are in the community, so the amount of patients with antibodies against these capsids increases as the patients are growing up. However, not all the patients with positive antibodies are excluded. We have set up a threshold, and we're using 2 different thresholds on how to get into the trial. One is for safety reasons, what we call the total amount of antibodies you may have against this viral vector. And the other one is for efficacy reasons. How much of those antibodies are neutralizing, meaning that they're not going to allow the virus to express the load, which is the dystrophin gene. Thank you. And the exclusion criteria are rather for you to be expected. Of course, the patients cannot be receiving another experimental drug, and if they are in a treatment that modifies the dystrophin expression, a wash-off period is allowed before the inclusion into the trial. So the last slide is to share with you where we are at now. We dosed our first patient in February this year. A second patient was randomized to the control arm. And we, as you may know, had a clinical hold, which is when the FDA asks you not to continue enrolling patients until safety is clarified, because we had an adverse event on this first patient. The event fully resolved. The patient is doing very well. Actually, throughout the course of the adverse event, he was mostly asymptomatic. And now the patient returned to his normal activities and continues to be followed per protocol. We put all of this together in front of the FDA, and the FDA has released the clinical hold, so we're now ready to resume the clinical trials activities at the University of Florida, which is our one site open, but we're opening other sites in the US and hopefully in Europe in the near future. So we have returned to our normal train of activities. I mentioned before that we're collaborating with Dr. Barry Ben and his fabulous team at the University of Florida, who provided excellent clinical care for our two patients in this trial. We're planning now to dose several children before moving back to adolescents. We continue with the objective of dosing the entire population of patients, now children and adolescents, hopefully in the near future to expand on both ends of the spectrum. And with that, happy for you to engage in a question and answer later on today. So thank you for listening, and happy to answer your questions.
Terrific. Thank you, Jorge. And thank you for putting some basic information in there. I should mention that PPMD has hosted several webinars on gene therapy, and you can find those on their website if you'd like more background information. We didn't feel like there was enough time today to put in a lot of background. Next up is Dr. Jerry Mendell from Nationwide Children's.
Thank you for this opportunity. And I think before I get started, I want to convey that this is not a competition between gene therapy programs. We all have slightly different strategies, but the results that we have at the beginning of our trial are optimistic for the whole field, irrespective of the slight differences that you'll see in transgenes that we use. And just another comment, I want to be sure that you understand that it was Pat Furlong who joined us in initiating this trial with donations that went to PPMD, and we could have never been where we are now. And then we are fortunately joined with Sarepta, who I've had experience with way back when they were AVI, and have had a very productive relationship with, including attempts at treating patients with oligonucleotides. So my partner here, as Doug Ingram told you, was Louise Rodino-Claypack. As far as losing her, yes, in some ways, but fortunately, we have an agreement with Sarepta that Nationwide Children's will be in close proximity to the new facility in Columbus. So I don't need to spend much time on this, just to point out a couple of things. You've heard why we have to use a modified transgene, and whether you use, in this transgene, we use one that was developed very early in the development of transgenes by Doug Harper and by Jeff Chamberlain, and it involves 4 spectrin repeats, S1, 2, and 3, and then S24 as the final transgene that you see. And the important thing here is a couple things more to point out, that we use an MHCK7 promoter, and you'll see the value of that. And then most of you are not familiar with AAV-RH74, and just to comment on that. We've used RH74 because there are less proprietary issues. This was a virus that was isolated at Children's Hospital, so we could step right into it. And we did many, many studies to show its value, both at it demonstrated efficacy in animals with widespread distribution and good gene expression. And we have gone on to demonstrate safety with RH74 in that we have had no observed SAEs with RH74, and we've injected 14 human subjects without any significant side effects. And we actually have some collaboration with the FDA. They've approved 6 INDs for us to use RH74. On the other side of the coin, I think what we learned, and I've been at this gene therapy for quite a while now. We did our first trial in 1999 and then rolled out series of studies. None have been as successful as this, but that's part of the learning process. But what we learned from our SMA trial, which was a very productive and beneficial program, we learned that what you see in the preclinical is what you hopefully anticipate in the clinical. And the one thing to point out there is in the gene therapy trial for SMA, we used the highest dose of virus for the clinical trial that had ever been used in clinical trial. We used 2e to the 14th vector genomes per kilo. And you can see in the animal studies that I'll show you, the numbers maybe aren't so important, but it was a dose escalation study with the therapeutic target at 2e to the 14th and one higher to see if we could achieve any greater efficacy. And basically we had, with systemic delivery in the mouse, and actually I'm not presenting data, but we also had it in non-human primates, we could show efficacy. We had expression levels with both 2e to the 14th and 6e to the 14th that we had 75% expression in all muscles that we examined. And the other thing we had that was very gratifying was very good heart expression. Actually, it exceeded anything we got in limb muscles. And this paints a very positive picture as we go forward. And this MHCK7 promoter is one that shares expression ability both with heart and skeletal muscle. And also, I think as you've seen in comments from these other trials, that we expect to have a dystrophin-associated protein complex that is reassembled, and that's exactly what we had in the preclinical. So we entered this with excellent experience in SMA. Basically, we save lives now for as long as 4 years in the SMA trial. And now we expected to have very good, or hope to have very good trial experience with DMD. And I have to say, DMD has been one of my passions since my early days, and I've probably been around longer than at least my colleagues up here on the panel. I first saw my first patient with Duchenne dystrophy in 1969 when I was at NIH, and I have firmly committed that I want to see changes in this field more than anything else. What we did was we planned an open label trial to initiate gene therapy with 12 subjects, and they were at 2 different age ranges for very good reason. One, the 3 months to 3 year of age trial was so we could treat patients without corticosteroids. And then, as brought up earlier by Stu Peltz, standard of care is steroids, one way or another, Emflaza or prednisone, and we had 6 subjects from 4 to 7 who were on steroids. This gives us the opportunity to see differences between gene therapy alone and the prednisone plus gene therapy. Right now, we don't know whether we need both or only one of the treatments. We hope to be able to find that out, and that'll be one of the targets of our program. For this trial, and I think it's interesting, and I'm so happy to see that these other trials are using a broader scope of enrolling criteria than we are. And the reason for this is from our past experience. We did a DMD trial back in 2006, published in 2010, where we did have antibody production when we expressed the gene in an area of the patient's deletion. So we have restricted initial enrollment. This doesn't mean forever. This means that we wanted to line up this study in the best possible format we could. We didn't want variables. If we had some patients with efficacy and some not, we didn't want it to be related specifically to their mutation. So this is a very homogeneous group. We also learned in our trials from previous studies that there were restrictions on the amount of pre-existing. This doesn't mean post-existing antibody. This is pre-existing antibody. And with Rh74, there's about a 15% incidence of pre-existing antibody to this particular virus. And so the FDA, working with the FDA in the past, we had 1 to 50 as the titer. In this trial, we actually have 1 to 400 as pre-existing antibody. Now, the FDA wanted us to start with the older group, as they often do. The logic you can challenge, but in any case, we started as they wanted us to, and we started with the older group. This was primarily a safety study, as first-in-human trials are often labeled. And we had secondary outpoints as biomarkers, which were change in microdystrophin expression pre to post therapy and decrease in CK if it occurred. And then we had a series of motor functions, 100-meter time test, North Star ambulatory are very powerful, and then a series of others, including cardiac MRI, because of our powerful MHCK7 promoter. Now, I show this because we haven't achieved this before, and I've done probably as many clinical trials as anybody in this room in terms of Duchenne dystrophy. And the one disappointment we've had over and over again, including steroids, although that's been challenged, but steroids, none of these really decrease the CK to near normal levels. And here you see CK levels at baseline anywhere between 20 and 35,000. And here's our results so far in the trial up to 90 days. We had an 87% drop in CK overall. This was the first reason to be comfortable with the trial with no safety issues. Then we also have biopsied the first 3 patients in this trial at 90 days. And we've had excellent gene expression that you can see here in the 3 muscle biopsies that we've done. And these are biopsies of the gastrocnemius muscle. And that is, I think, shows you that we can get the gene well distributed and into one of the muscles that's actually been charged to be most affected in some courts and less affected in others. And the less affected is in my particular court. But here's what we see: robust gene expression in the first 3 patients. We have gene expression with a mean of 76%. That's when we count individual muscle fibers in the biopsy and we show gene expression. And equally important is how much dystrophin is at the membrane. It's one thing to show positive fibers. It's another thing to show intense gene expression at the fiber. And you can see here that we have very similar results for mean intensity. And Western blots, somebody was explaining earlier, this is a hard concept often, but one that the FDA likes to use for gene expression. And at our Western blots at Nationwide, we had a 53 or 54% gene expression by Western blots, and those are framed there next to the pre-existing pre-existing dystrophin expression, which was minimal. And this was working with Sarepta. We have had vast experience in terms of reaffirmation of the Western blots. They do them with excluding fibrous tissue and fat. I'm sorry, including fibrous tissue and fat, and ours is excluding fibrous tissue and fat, and that's how we think of the difference here. We think ours is more accurate because it only measures what's in the muscle fiber. And the other thing that's important about this trial so far is that we get very good copies per nucleus of the gene that we put in. And don't forget, these are random samples. So we have gene expression. Your target gene expression is to have 1 copy of the vector genome that we put in per nucleus. And we have really achieved that level of expression. And also, again, I mentioned and others have that we want to see restoration of the dystrophin glycoprotein complex. And here you see two of the primary confirmatory proteins in that complex, alpha, I went backwards, alpha and beta sarcoglycan. So that all looks very favorable. But nothing looks as favorable as this, because we've been accused of toxicity in this whole field. Not only me, but the whole field has been accused of, do we have a toxic agent with AAV at these very high doses? And this is one of the patients who we did. This was immediately post-gene therapy, and we see a very happy, unaffected child. The parents are in the room when we inject the gene, and there's virtually no problem. And his parents are here, and I'm very grateful to them for being here, along with other parents who've been in the trial. The other thing is, this is our patient here. This is an outcome at about 2 months post-gene therapy, improved in the 10-meter walk, stair climbing, North Star, and I love this picture of him on his scooter with a drop in CK from 20,000 to 2,000. So very gratifying early results. And this is the same patient now who's ready for a soccer or rugby game, whatever you like most, and he runs very well with good proximal and distal muscle extension, proximal especially. We did an ILI study, an infusion study, in the, as Kevin explained to you, and one of the problems with that method is that as he showed, we get good quadriceps expression, but we don't get much expression in the proximal muscles. So as he said, he's moving on to systemic delivery, and we went there because of that. And this is one that I think is very good, and thank you for doing that. And you can see this was 2 days post-gene therapy on your left, and now this is at 60 days, 2 months post-gene therapy. The other one I said I don't know whether it said 2 days or 2 months. The one on the left is 2 days, the one on the right is 2 months. So we're getting good expression and at this point, and we've had no serious adverse events. 3 subjects have had elevated GGT, but we've seen that repeatedly with gene therapy trials. The first time I ever saw that was in our SMA trial, and I went immediately to the FDA and talked it over with them, and they were not the least bit concerned about liver enzyme elevations. No other clinically significant laboratory abnormalities. We did have 3 patients who had nausea early in the trial, and one thing about our trial, I think we can attribute that to steroids, most likely, not for sure, but most likely, because all of our patients in this trial are on weekend dosing of steroids, and I'm a big advocate for that to cut down side effects and promote long limb growth. And these patients now in this trial, what we did was we switched them, let them stay on their high dose on the weekend and switched them to a daily steroids during the week. High dose on weekends and daily. So it's quite a steroid load, but we had experience where in a previous trial where daily steroids to prevent the liver enzyme elevations seemed to be the right route of delivery. But don't forget, we have so much to learn that we are just embryonic in this field. We're pioneering everything, and Solid and Pfizer are along with us. And if we have success in all those trials, we'll be able to reach more patients. And like I said, I'm very thankful that these other trials are using a broader spectrum, and that will make many of you happy because you'll have the opportunity. So our trial is very restricted by age and by mutation for homogeneity purposes, but once we see that, we can begin to extend that. And I think the first thing we'll extend would be the pre-existing, the mutation, the patients who receive mutations will go beyond 18 to 58. And in summary, we've had consistent good preclinical results that predict a good outcome. We have upregulation of dystrophin-associated glycoprotein complex, reduction in CK, good vector genome copies, and the use of MCK promoter is very hopeful. And one thing I can say about that is my hope for the MCK promoter and working with Sarepta, I think will give us the opportunity is we're often asked, what about boys who are in wheelchairs? What about the 13, 14-year-old? You're biasing your studies to select a very targeted group, and all that, that's true. With an MHCK7 promoter, and the other researchers, I see Dong Sheng is in the audience, can speak to this. We have good gene expression in the heart, and if they have equally good gene expression in the heart, I think that we can take a 13, 14-year-old and really get very good gene expression in the heart and perhaps change their whole outlook and their whole prognosis for the future. So I want to thank you very much, and I appreciate the opportunity to be here.
Thank you, Jerry. Terrific stuff. And now, Tara Moorhead from Pfizer.
Thank you. Thanks, Dr. Cripe. Yes, my name, let me just get this going first. Okay. Yes, as Dr. Cripe just mentioned, my name is Tara Moorhead. I'm a director of clinical research at Pfizer, working exclusively in DMD, and feel very honored and humbled to be here, especially with such an esteemed panel. But also, I'm excited to be able to have the opportunity just to talk a little bit about Pfizer's first exclusively run gene therapy study, and that's a safety study in Duchenne. And just a disclaimer, as a Pfizer employee, in terms of any forward-looking statements I say today about this program. So just to get started, we have PF 069-39926 as the name of our gene therapy at this point. I don't know why we do that. It really rolls off the tongue. It used to be called BMB-D001, which isn't much better, but this was the name it had when it was owned and developed at Bamboo Therapeutics in North Carolina by some brilliant minds, Jude Samolsky, Zhao Wan Li, among many others, with seed funding from Cure Duchenne, one of the advocacy groups. And we acquired this company in August of 2016. We were really excited, but they developed a mini dystrophin transgene, and you hear micro, you hear mini. In my mind, I just think, to simplify, I think it's more tomato. Really, it's just a preference or terminology. But this gene sequence was based on a mild Becker patient's gene sequence, and it was a more mild form of muscular dystrophy with Becker, but even that patient, in terms of his clinical manifestation, it was a more mild phenotype or manifestation of the disease. And you'll see here, so we tried to, Zhao and team tried to pick the domains that they thought would be perfect and that would fit, as Dr. Quiroz mentioned, would fit in a capsid that could also include as well a promoter and a muscle, and be codon optimized to really get the gene where it needs to go. And our serotype is different from Dr. Mendell's, the capsid that we're using, but we believe that it'll have global transduction and expression, not just in the skeletal muscles, but also in the heart. And so speaking of the heart, we used, just before we get into the clinical program, just to spend a few moments talking about our preclinical data that really got us excited and gave us the confidence that we had something to move forward with. We used a DMD MDX rat model, and that's a little different from what you may have heard in terms of the MDX mouse. And we picked that model for many reasons, one of which was these rats developed cardiomyopathy, and we knew that we would see transduction in the heart, so we thought this would be a good model. And we did indeed see that, and I had a slide, but I was afraid I didn't think we'd have time, but we saw great expression, even in the heart, in terms of positive muscle fibers. At the higher dose levels, we were seeing over 85 to 95% on average in those groups. But other data that we saw, there's been a lot of buzz about CK, and so I just thought I might share. We looked at 2 different time points for CK, and these data were shared at the World Muscle Society meeting this past October, and then also at the PPMD on this meeting this past February. And so you'll see in the first kind of column, I don't have a pointer, apologies, but the wild type or the control rat, you'll see the CK was low. And you'll see in the 2 higher dose levels, and that's 1e to the 14th vector genomes per kilogram and 3e to the 14th vector genomes per kilogram, you'll see that at both time points, the CK had dropped and stayed low. So you'll see that dose-dependent decrease. We also looked at some functional assessments, and that's similar to what I think has been described in terms of grip strength or force and repeated, so not just what the initial grip strength is, but also with repeated measures to measure the fatigue and the sustained force. And you'll see here, and the same 2 dose levels that I just described, the little black line with the little black boxes would be the control group, so that's that line that consistently the force grip strength is consistent. But you'll see that there's a DMD MDX mouse rat that wasn't dosed with anything, and that kind of dropped off. That's the little black circles. But for the 2 dose levels that I just discussed, you'll see that the force grip strength over time or repeated maintained or even was better than the control rat. So that got us pretty excited, as well as the safety that we saw. And so we've now entered into the clinic, so I'll take a couple minutes just to talk about that. This is a phase 1b study, so safety study is our primary objective, but we are also looking at a couple other endpoints, and this is our first in human, first in patient, so we went straight to patients with DMD. Our patient population is 5 to 12-year-old boys with DMD who are ambulant and on daily glucocorticoids that can tolerate the daily glucocorticoids. And we are also looking, as mentioned, in terms of making sure that they don't have the presence of the neutralizing antibodies to the AAV9 capsid. And additional inclusion-exclusion criteria can be found on the, there's an NCT number at the top, and that's on clinicaltrials.gov. This is a single intravenous infusion, as explained, that once it's introduced, you will develop the the neutralizing antibodies. And so we want to make sure that the reason for that, we picked 2 dose levels that we believed would be therapeutic, and that either of which, or we're hoping will be therapeutic, either of which may end up being the dose that's selected to go forward with in terms of commercialization, should we be so lucky to get to that point. And you can guess what they might be. But we're looking at about 6 patients in each cohort, and we are staggering the dosing just for safety so that we can monitor, so at least 6 weeks between the first 2 subjects in each cohort, and then provided we don't see anything, we can then dose at a frequency of about 3 weeks. We have an external data monitoring committee, or you might have heard a DSMB, or Data Safety Monitoring Board, and they're just an external group of experts that will just help keep us objective in terms of monitoring safety and whether we can escalate to the next dose level. As I mentioned, this is a safety study, so that's our primary focus, and we're looking at that through 12 months. But we're also looking at dystrophin expression and transduction, and so we're also taking 3 biopsies, 1 at baseline, so we have something to compare to. Another at 2 months, where we expect to see about peak expression, and then because we're expecting that long-term persistence, again at 12 months. And because we do expect long-term persistence, or we're hoping for that, we are also monitoring for long-term safety and just to see how the boys progress over time, and those would be for additional 4 years. But appreciating the trial burden, those will just be annual visits, so hopefully not too bad. And so just where are we? We are still very early on, but just quickly, we have 3 sites in the US. This is US only right at this point for our 12 subjects. We've screened 4 subjects thus far, patients I should say, one of whom was a screen failure, unfortunately, due to the ambulatory criterion. But we have enrolled 3 patients and dosed 3. And this is just a very basic graph, just to give an appreciation for how far we are. So our first patient was dosed in March. We had a press release about that, and he just completed his 3-month visit this past week. And our second patient completed his 1 month just earlier this week, and our 3rd patient just was a week out as of yesterday. So we're very excited, but now we're at a point where we're just having to be patient and allow the data to come in so that then we have something to interpret. But as far as the safety is concerned, so far, so good. It seems to be well tolerated, and so we're excited about that. And so just to talk about manufacturing, because that's another thing, a big consideration when it comes to gene therapy and how difficult it's to manufacture. When we acquired Bamboo in 2016, we also were fortunate not only to get the assets and the expertise, but also their vector core facility for manufacturing, which has been so helpful. Under the guidance of Josh Grieger at Bamboo, we were able to really get the process in really great shape, and it's this vector core that's supplying all the material for our current trial. But then Pfizer really made a determination that they wanted to own the manufacturing process and have that control and be able to get it to where it needs to be in order to support the large amount of virus, or large amount of material, I should say, for diseases like Duchenne. So in August of last year, we decided to invest $100 million in one of our other existing facilities in North Carolina to really get it so that should we be so lucky as to get to the point where we can commercialize this material, we get approval, we can actually meet the demand, and we can supply what needs to be, because I think that's also something we really need to be thoughtful about. And so I think I'll just end with a couple thank yous. As I mentioned yesterday, we were fortunate enough, thanks to the help of Catherine Beaverson, to meet with a number of our the advocacy organizations who were gracious enough to meet with us to help us in terms of different aspects about our clinical trial design. So many thanks to them, as well as some clinicians, both in the US and the UK. And yeah, we're just grateful for helping us stay humble and be informed of what matters to the patients. And then, of course, so thanks to all the advocacy groups, the investigative sites, last but not least, the families, especially the families, as we're really, as Dr. Mendell mentioned, we're embryonic in this phase of development of gene therapy, so the courage that it takes to be able to really help us to advance the science and to be at this point, we're so grateful. And of course, thanks to the other researchers too that keep us motivated as well in terms of really trying to get what we need and what we're hoping for is transformational therapies for this patient population. So thanks so much.
Okay, so we're going to, I'm here as a timekeeper. We're going to reinstigate the discussion about gene therapy, and we've got a couple new panelists from, although speakers from earlier today, but just to remind everybody who people are, we'll just go down the line and introduce ourselves and then launch into it. So we've already got lineups at the microphone. So just to recap who we are, why don't we start on that end and introduce yourselves for the audience.
I'm Doug Ingram, the CEO of Sarepta Therapeutics.
Tara Moorhead, Director of Clinical Research at Pfizer.
Jerry Mendell, Nationwide Children's Hospital.
Kevin Flanigan, Nationwide Children's Hospital.
Kyle Mara, CSO of Solid BioSciences.
Great. So why don't we start, and we'll just alternate side to side. Let's start on my right.
Good afternoon. Again, thank you all for your presentations earlier today. A question regarding the AAV virus and how some boys will have that and some of the gene therapy wouldn't work for them because they've been exposed to it. Just some explanation on how those boys would have been exposed to it. Would it have been through other medications that they would have been on before, or how would they have had that exposure?
So several of the AAV viruses, they're in the population. They're just out there in the environment. So exposure can be passed just in growing up, just in environmental exposure itself. It's not as a treatment option. These are not capsids that have been used for therapy so far. Environmental exposure can happen with, we know that the rates of antibody positivity go up with age, so that's a signal that it's just exposure within the environment. These don't cause any illnesses, so they don't cause symptoms when children are exposed to them or when people are exposed to them.
Sort of like catching a cold in many respects.
So is there something that we should or we shouldn't be doing with the boys to, try to have them least likely to be exposed to any AAVs that would limit their ability for any trials?
So it's a question in a way, should you quarantine your child really from the environment for it? And I think we all agree probably these are kids and they're boys first before they're boys with Duchenne, and avoiding school, avoiding socialization, and all of those things is really probably to their detriment. So I wouldn't recommend it. I don't think
we don't even know if that would work. Right?
We don't know if that would work.
These are subclinical infections.
Okay. Thank you.
Next question. my left.
Okay, so first off, I just wanted to thank all of you for all of your hard work and dedication towards Duchenne muscular dystrophy. But my question in particular is for Dr. Mendell. So so far for your clinical trial, all the boys have been ambulatory. So my question is down the future, do you plan on having clinical trials for non-ambulatory boys as well? And if so, how would you expect them to respond to it? Do you think they would regain any mobility by any chance, or no?
The last part of the question about increasing mobility, I'll reserve. But as far as treating the older boys, absolutely. We definitely have a program, and Doug Ingram is here, and we've talked about it together. We have a transcript or a gene construct. That's the thing we showed you, and it has a heart promoter, and that heart promoter will allow us to deliver the virus and have a good bit of it go to the heart. In boys who are non-ambulatory, that would be a major change in their natural history. At the same time, I envision that even if they're far advanced and they don't have much leg function, we would be able to save arm function. So I think the possibilities are there, and we're very eager to move ahead with that.
Okay, thank you very much.
Any comments from the others about that?
So Solid has a non-ambulance in the study design. As you know, we're moving to the children for the next couple of patients, but we're hopeful to get back to the non-ambulant population as soon as possible.
Well, I guess we'll go back to the other side, but we'll do two on each side at a time for the sake of the cameraman's sanity.
I just wanted to thank you all for giving us so much hope, to our boys and to our families. I have a few questions myself, and then a few questions from families who are not able to be here today. So my first question is to Ignite, to Solid. You said that you have plans to open up sites other places besides Florida. Do we know what sites they are? And also, the other question is, if you have good results, preliminary results, will you be letting us know about that in a few months' time?
So in regards to the sites, we haven't finalized contracting, so we can't comment, but they'll be on clinicaltrials.gov as soon as possible,
Okay.
And that will be available. In terms of disclosing the data or reporting out the data, we will wait until we have a good, robust data set, and then share that with the community.
Okay.
Are you looking to
Thank you.
spread those sites geographically around the country or
Yeah.
other countries? Or?
Yeah, we're hoping to spread it around and also move into the UK, Europe as soon as we can as well.
Oh, okay, good. My next question is to Dr. Mendell. So when do you anticipate doing trials for older kids? Is it going to be a concurrent trial, or are you going to wait to finish this trial first?
Well, I think that our first step will be to expand this trial, roughly looking at the same age group to confirm. This is a pilot trial. We'd want to
Right.
confirm that, and then we would move on to older boys.
Okay, thank you. And then also, this question is for everyone. When do you plan on expansion to Europe and Canada? And also, any trials for manifesting carriers? Do you have any plans for that? And also for rare mutations. I think Dr. Mendell touched on that a little earlier, but just briefly, you can just.
So as I said, we're hoping to expand as soon as possible into Europe.
Right.
And then the other one was we're in the non-ambulant population. What was your other question?
The
Sorry.
manifesting carriers, is there anything in the future?
We honestly haven't thought about that too much, but it's something that's coming up and we should address and look into. And then there was one other question, wasn't there?
Okay.
Rare mutations, we have no limitations on the mutations coming into the inclusion,
Right,
into
okay.
the study.
How
Others
about the
want
others?
to answer that question? Kevin or Tara, any comments on your trials about those questions?
Actually, we have the same plan as Carl just mentioned, so it's in terms of expanding, we hope to as soon as possible. Mutations, we're not specific, at least for this first trial, we're trying to just make sure that it's a mutation that we know manifests as Duchenne, but we're not looking for only specific mutations.
Okay, and the manifesting carriers?
It's something that I guess we need to explore a bit more as well.
Okay, but there's nothing right now in the...
No.
Okay. One more question. Yeah, and with the steroids, like the inclusion criteria is, I think Pfizer, you said it's like daily dose?
Yes.
Versus, how about the others? Is it still the daily dose or weekend dose?
So it's a standard regimen,
Right.
glucocorticoid, and then we modify once you're
Okay, but it
recruited onto
could
the
be
trial.
daily or weekend.
As far as I know, yes.
Okay. And then also, my last question, sorry.
That's okay. We gave
I
you
know
lots
you've
of time to
been,
write it down, so.
yeah, these therapies have been addressing like the muscle and the cardiac muscle cells. How about those that address the satellite cells? Is that something in the future that you'll be looking at?
Well, you hit on a very critical question, actually, and we are looking at that in our current muscle biopsies. And there have been reports from other delivery systems where satellite cells are transfected. So I think it's extremely important for long-term expression.
Right.
So yes, we're looking at it.
Okay. Thank you.
Great questions. Thank you. We'll stay on that side for one more.
Thank you. So this is for the community at large, and Dr. Mendell and anybody else that would like to chime in. I believe I heard either during R&D Day or maybe PPMD gene therapy webinar, if you could clarify that gene therapy may not be for 100% of patients. I believe that there were some mutations that this would not help. Can you clarify that for the community?
Well, I'll clarify that because what we found in our first Duchenne trial is when we expressed the gene in the area of the patient's deletion, they produced an antibody. So we framed this trial, the current one, this pilot trial, to avoid that problem. But because we wanted one, homogeneity in the population, we didn't want to have a variable produce a false negative response that would go outside the gene delivery issue. And so I think in a pilot trial, what you try to do is to line up all the cards, so to speak, in a way that will give you the most reliable answers. But having said that, I feel that we have the possibility of going outside that. And we showed antibody, but we really don't know its clinical manifestations. We don't know whether it really, that antibody that's produced after the gene is expressed, whether that has anything to do and that will cause an inflammatory or a T cell response invading the fibers. So I think we'll do that very shortly, but I think we'll have to frame it in such a way that it will be a unique cohort so we can answer a question. If you try to answer too many questions in the same cohort, you're bound to fail because these gene therapy trials are relatively small. They don't have the power of studies that we do in the pharmacologic industry. We have to frame it so we can answer the question specifically and then start moving from that point forward.
So would anyone say that there would be exclusions at this time, or is it too early to commit to that?
I think the other trials they can answer. I don't think they're excluding. And frankly, I'm very happy about that because this gives us the opportunity to do one trial where we framed it in such a way, and we'll see other trials where we'll be able to answer the question and compare. And that's absolutely perfect in this community.
Okay, thank you. Anyone else care to chime in?
Yeah, so we do not exclude based on mutation.
Okay, thank you.
Okay, switching sides.
I have a couple of what I hope are very quick questions for everybody. Specific for Pfizer, I have 2 questions. Can you talk a little bit about what your dose is and your plans to escalate it? And then also, as a mom of older boys, I'm very excited about potential heart expression. So if you could talk a little bit about what you're seeing in preclinical in terms of heart expression.
Sure. So as far as our dose levels, kind of what we showed for the DMD rat, we did go with the 2 highest dose levels that we had shown. That's 1e to the 14th vector genomes per kilogram, and then 3e to the 14th. And both of those seemingly could end up being the therapeutic dose. So we thought we were playing it safe and seeing what we saw, and if we saw safety in one versus the other, that's what. But we're starting off with the lower dose level, and then just looking to escalate to really get clarity in terms of if one seems better, one seems safer. As far your second question was the
Heart expression.
The heart expression. So I didn't get a chance to show it. I wish I had, but in this DMD rat model, we saw great expression. If you think of the wild type or the control would have 100%. We saw in terms of positive fibers in those 2 cohorts, actually they weren't even statistically significant from each other, so that they were that comparable, and they were in the over 80 to 95%.
Oh, that's great.
Yeah.
Question for Solid that you may or may not be able to answer. Have you worked out with the FDA how you're going to escalate the dose and get back into the older guys, and what type of criteria you're going to use to know how and when to do that?
So that will be decided by DSMB as well. So we put a proposal in front of the FDA, and they're agreeable to that. As we go into the kids, go into a set number of kids, and then at a point we'll reevaluate, and with the DSMB approval, we can move back into the non-ambulance.
Thank you.
Drug Safety Monitoring Board. Sorry.
And then just one last one for everybody. I'm hearing a lot of chatter about screening failures, and I just wondered if you could speak to what the numbers look like for the amount of screen failures due to antibodies in these guys.
So.
Oh, go ahead.
So we don't sort of talk too much about screen fails, but it's what we anticipated based on some of the data coming out of from Kevin and others in terms of antibody levels.
So can I speak to that? So about 20% is what, about 20% you'd say? Yes, and we see the same. And then granted we've screened 4 patients, but hopefully that will continue and maybe even be better. Yeah.
And actually comment in the future, there are ways to remove those antibodies temporarily, like plasmapheresis, and maybe those kinds of things could be, included in future studies.
Yeah, thank you.
More patients. Next on that side.
That was a perfect segue to my question. So I hear a lot of talk about ways we might mitigate this immune response in the boys that are having So could you talk a little bit about the 3 companies and what they're seeing and how soon you might get something like that underway?
Let me start by saying that I think to avoid confusion, we should clarify that every boy who gets a virus is going to develop a serum antibody, period. And that serum antibody is post-gene delivery. It will have very little to do with gene expression at that point. What is the critical issue is, and we've seen this in clinical trial, reported it in clinical trial, and that's how we got to the titers that preclude patients. And we saw blocking antibodies in our LGMD 2D trial at 1 to 1600. So that patient did not express, and we could see multiple confirmations of that. So that's where the pre-existing antibody, pre-existing, post-existing. Pre-existing is important, post-existing by all criteria. If you look at the numbers, they'll blow your mind as to what kind of antibody levels you get post. Now, the other factor is T cells. We also think that pre-existing T cells to viral capsid are not favorable for gene expression. Because if the T cells are already there to AAV capsid, and we've seen this as well, if you deliver that AAV capsid and you have T cell response, but when that AAV capsid is delivered, you're going to have T-cell invasion of those fibers, and that's going to preclude your gene expression. So there are differences in the immune response. There are T cells, there's pre-existing, there's post therapy antibody levels. So I think that actually Tim was just saying we've done one experiment where we did see pre-existing antibody to AAV, as was in AAV8 as it turned out, because we were looking at non-human primates. And we did an experiment and published it where we could clear the system of AAV8 antibody and have about a week's time to deliver the gene. And frankly, I'm itching to do that because we have boys who have had AAV previously, and I think I would like to give them the opportunity to have the new things as they're coming along, and I think we'll be able to get there. But we need funding, and actually Sarepta is helping us move in that direction, and also that's the issue for redosing too.
Does that introduce an element of risk, additional risk?
In my opinion, it does not, but opinion isn't worth much. It's what happens in the clinical trial, and I don't know whether Doug wants to mention anything about supporting those kinds of efforts for redosing.
No, I mean, it is I think from our perspective, just as Dr. Mendell said, if we're fortunate and the gene therapy program works as our preliminary results give us hope that it will, the very next thing that we have to think about is redosing, because it's not just redosing, it's topping up in the future, it's dealing with boys that have neutralizing antibodies coming in. The good news with our current program is that the screen fails rate is very low right now. But
isn't that in young boys though? I mean, the exposure is going to go up as the boys age. So
I believe we have some data in older boys as well, but it does get to the point that we've got to deal with this issue, and we're gonna start that process from a research perspective this year. So we're not going to wait post-approval for it. So we want,
that was a
Dr.
good
Mendell
answer.
said he's itching to get it done. We're itching to make sure that we fund it properly to get it done.
Yeah.
So it's
And just to answer, so Pfizer's also looking at preclinical, looking at monkeys to study immunomodulation to neutralizing antibodies and what we might be able to do to overcome this challenge.
Is there a time frame on that one?
We're still a ways away because I think we have to understand it and then make sure that we think it's going to be applicable and also going to translate well in humans. So I think it's going to take a little bit of time for sure.
Okay,
but
thanks.
it is something we need to plan for.
I haven't forgotten about you, Solid.
No, thank you. Yeah, so we also have active programs to look at pre-existing antibodies and identify methods to get around that. And then also importantly, thinking about re-administration down the line as well. Are there other methods and things we can do to help allow for that in the future?
And these studies have already started. We're not going to be doing them 2 years from now after we see what we think we're going to see in these first trials.
Well,
it's going to be concurrent.
Yeah, so we're hoping to, because if as Jerry mentioned, this could be hopefully put in front of the ongoing studies if it's identified as a way. It's a technique. It shouldn't really change and require future studies if this is just a plasmapheresis, for example, that you put in front of the dosing.
Great, thank you.
It's never fast enough, though. One more on this side, then we'll go to Kathy.
In continuation to what she was talking about, that if we have a immune reaction or a NAB against the AAV9, is it possible to switch over to another virus with a serotype, different serotype, which may not have a immune response?
So there's likely to be some cross-reactivity, but that is an option, yes. That could be a potential option.
Yes.
Hi, my question is for Sarepta and Pfizer. So if these clinical trials are successful and you go for an FDA approval, do you think you'll be going for a broader label so that right now for Pfizer, the inclusion criteria is boys between 6 to 12, and then for Sarepta is 4 to 7, and then there's mutation limitation. So if you go for approval, are you guys going to go for a broader age label mutation?
Yeah, well, the short answer is Dr. Mendell said is we are absolutely, our goal is to have a program that gets to a broad indication, and a broad indication, you know, the broadest possible as we track in. So what Dr. Mendell is doing is being very thoughtful about the approach so that we don't do everything and then we fail as a result. So as Dr. Mendell mentioned, it's the current thinking at least, and we've got to talk to the FDA, by the way, about our clinical approach. So we'll have a meeting in the coming few months to really discuss our clinical approach with the FDA. But among the things we're thinking is we would be looking at a narrow cohort of age right now, but then we would separately do a cohort to look at older children and adults, look at younger children, do some additional work and start getting comfortable around the exons that are below 18 as well to deal with that issue. And so, you know, our goal again is not to get a narrow indication. Our goal is to start with a trial that is properly sort of, you know, risk adjusted so that we, you know, can be successful, layer in additional cohorts along the way, and then find ourselves in a position where we can seek a broad label, get it to the community in the United States, Europe, Asia Pacific, Africa, Middle East, everywhere, South America.
Okay, thank you.
And likewise, easy answer, we're taking the same approach.
So you guys plan to do it in boys above 12 and below 6 before you go for an approval?
We might do it in parallel, actually, and sometimes, and it's also a discussion that we have to have with regulators to see what flexibility, if we establish safety, you know, what flexibility they'll allow for a broad label and how much, what if anything we need to explore in certain age groups for them to feel comfortable with approving as broadly as they can. You know.
Okay. Thank you.
Thank you.
Okay, other side. Kathy.
Hi, this question is for Dr. Flanigan. So, Kevin, I know that now regionally you're providing GAL-GT, well, you're providing GAL-GT regionally with plans to infuse systemically. So could you tell a little bit about what the process is, what you need to prove with the regional delivery prior to going to systemic infusion, and what your timeline is, and if you have an exclusion criteria for those patients as well?
Sure. So GAL-GT2, the trial was originally designed some years back, actually beginning with intramuscular injection, then moving to local, regional, and into systemic. That was the pathway, the trail blazed by Dr. Mendell and other studies. The ground has shifted a little under our feet as we've done this. There's much more experience with AAV delivery in patients, and right now we have sufficient virus to complete additional patients. We'll need to continue with synthesis of more virus to do a systemic delivery trial. We anticipate, this is a licensed technology to Sarepta, and we anticipate that we will be successful in manufacturing enough that to continue this in the near term. There's decisions involved at Sarepta's level about that, but we don't have I anticipate that protocol will follow along with ambulant boys in the model that we've done. We have a well-established protocol. For all the reasons you've heard here, we're going to initially do systemic delivery in an ambulant population. We'll have to see what the FDA says about our final design of that protocol that has not yet been finally designed.
Okay. And exclusion criteria?
Pardon me?
Any exclusion criteria for those patients?
Oh, we don't anticipate exclusion criteria based on any mutation class. We do anticipate it will be ambulant boys, and right now we don't have exclusion criteria. We haven't finished the design of what will be the systemic trial for that.
Great, thank you so much.
Yeah.
So are there certain mutations that would be more likely to have an immune response with the T cell issue?
You want to take that, Jerry?
Well, it's a tough question. I don't think we really know the answer to it. You're talking about pre-existing viral antibodies or viral T cells, and I don't think that's pre-existing. There might be a possibility that the gene mutations in the more at the amino terminal, since they are so devastating and beyond what you might expect compared to, let's say, mutations in the central part of the gene, there might be more pre-existing response and the biopsies tend to be more inflammatory at the amino terminal. So I don't think we can answer the question, we can only speculate. It just hasn't been studied to the degree that you're asking. But the important thing is that when you design a clinical trial, that you put into your protocol that you'll look at that pre-existing T-cell response before you do it. But it is a good question, and I think over time we will have more data on that.
And is it something that can be remedied at some point? If it looks like there is
You're asking the wrong person. That's the person up there to ask. He's our immunologist, and he can answer the question.
I mean, maybe, yeah. There are lots of ways to reduce immune responses, and we do a lot of that. But it's always a balance of pros and cons of doing that. I think one concept that's important that maybe hasn't been made clear yet is, the virus is this shell of proteins that's encoding the gene. And the reason the pre-existing immunity is unfavorable is that inactivates that shell right away as soon as you infuse it into the bloodstream. But once the virus has delivered the package, the DNA, into the cell, those proteins are no longer being produced. They're going to be broken down by the cell and presented to the immune system transiently, but eventually they're basically going to go away. So if you can get through that period of time that, without destroying those cells that contain the gene, then you'll probably be in the clear, because the only thing left then would be an antibody or immune response against the foreign part of the Duchenne, of the dystrophin gene. So I think, there's the possibility, but if you need to suppress immunity, there's a lot of ways to do it, but again, they have their own risks, because then you need immune system against infections too. So.
Okay, so there would be hope for those that have the pre-existing issues.
We always have hope, yes.
To be able to still be able to introduce it to
There's
them.
hope.
Okay, thank you.
Back to this side.
Hello, everybody. Thank you for being here and for the wonderful presentations. Back to the satellite cell transfection. Thank you, Dr. Mendell, for speaking to your gene therapy efforts. I was hoping to ask the same of Pfizer and Solid, and a couple of things. Was wondering if that's also a characteristic that you're looking to record. And in addition, is this something that's included in the 10-year effective measure that you're discussing, or if this is exclusive of that?
Before the answer, I think we need to end. No one else come into the line, because we got 1 more minute left. So we'll finish off these 4 questions and end. Go ahead.
You going? You go. All right. So, both Pfizer and Solid are using AAV9, which was shown by a lab at Harvard that it actually gets into satellite cells. So there's a publication. We haven't looked for it specifically ourselves, but there's a publication indicating that it is able to get in there. We don't know what that means, and I think as we go forward, we'll better learn what it actually means in terms of allowing for durability of expression going forward.
Thank you. And very quickly, the other 6 vectors that have been approved, are they specifically approved for microdystrophin or minidystrophin delivery, or are they not candidates for that?
Well, just to comment, I mean, approval depends on preclinical findings using a specific serotype. So any potential serotype, we have 100 serotypes of AAV, but the most common ones are 1 through 10. And any of those, and some of them express better in muscle than others. So you have to, this is where I was trying to emphasize, probably not enough, but the preclinical is critical in terms of predicting the clinical response. And that's how we got to this level of dosing, and that's how we got to showing the FDA that not only was the preclinical effective, but then the FDA demands then that you do an extensive toxicology study on the GMP product, the product you're going to take to the clinic, and make sure that there aren't any toxic side effects. That's kind of the paradigm that works.
Thank you.
Okay, third to last question.
So, question is for Dr. Mendell. If we take a gene therapy, then in few years, say if gene therapy is not effective, would we be able to go back to PPMO or PMO therapy? Any possibility, or that's out of the questions if we take a gene therapy?
I don't see any contradiction to that. I think you could do that. If you were, I mean, as we move ahead and become more knowledgeable, I think what the ideal, let's say that you found an antibody to AAV that you have designed for this clinical trial, and you have a mutation that would respond to 51, 53, or 45 skipping. The smart thing to do would be to start with skipping so you don't produce, have any antibody interference, and then later on see what the results are, compare it with gene therapy, and then design the clinical trial depending on the response. But it'd be better to go the other way if that was your option.
Okay, thank you.
And I'd just like to add to that, so just a clarifying comment that it is possible that if you've been exposed to gene therapy that you might not be able to participate in other clinical trials, just because if you do have that long-term persistence, it might interfere with what you're seeing in terms of the clinical trial itself. But I think I agree with Dr. Mendell, for approved therapies, I don't see why you would be precluded from taking those down the line, and that could help maybe enhance the benefit that your child is getting. Is that fair?
Thank you.
Sure.
Second to last.
Thank you, everyone, for the work that you do. So there is a lot of excitement and hope and promise. I'm sure there are also a lot of challenges. So I'm wondering if each of the trial could talk about, one, like the biggest challenge that they can anticipate at this point.
Down the line.
All right, I guess I'm first. I think manufacturing is going to be a big challenge. And I think that's part of what's great to see here is you have three companies both going after this, or all going after this. And so hopefully we'll be able to find a solution and be able to support treatment for the entire community. You think if you start doing the math, 1E14 vector genomes per kg multiplied by the average kilogram weight, and then how many patients there are, the number gets big really, really fast. And I think we need a lot of really smart people to be thinking about this now as we move forward.
I can't help but add this since I'm very emotional about this whole process. Look at the SMA trial that we published. 2e to the 14th vector genomes. Kids die between 8 months and by 2 years of age, there is more than 90% mortality. Every life in that trial that we published has been saved, and now saved for up to 4 years. Now, there are differences between trying to transduce a multinucleated muscle cell and a single neuron with SMA. And I think there was a time, actually, when we would have predicted, all of us who are doing translational therapy, that the SMA success would have followed the muscle success because of delivery. It was the blood-brain barrier that was the most I think the most frightening in terms of delivery, but think about how well that trial went and how many lives were saved and how much function, and I think you'll have a perspective, at least for some of us who have been in this business a long time.
Kevin, challenges?
I was going to just agree that manufacturing is a challenge. We're fortunate at Nationwide Children's Hospital to be affiliated with a GMP production facility that has been really central to the success of the trials at NCH and trials elsewhere, in fact, for manufacturing of GMP, GMP quality virus. But that's really scalable at present scalable to the phase 1/2 trial. Now, I agree, all the companies here, and I'm sure Doug will comment on what I could say, are working really hard to scale up manufacturing capacity. That's going to be the bottleneck, I think, for many of these therapies.
Tara?
So, as I mentioned, we're working really hard in getting in position in terms of our manufacturing capabilities in-house. I think, in addition to that, though, what we worry about is just making sure that we can demonstrate the clinical benefit, given that the disease progression is a lot slower than it would be, for example, in SMA, and just making sure that we have robust data set as quickly as possible, right? To support that it's not just about expression, we also want to be able to show that we're actually improving the lives and the quality of life and the health of these boys.
Thank you. Doug, challenges?
Well, I mean, look, the challenges are obviously the sciences, but as Dr. Mendell says, so much has been answered, so much more remains to be answered, but we have a lot of hope. Certainly manufacturing is an enormous issue for us. We have on our side, we have a relationship now with Brammer BioSciences. We'll continue to have additional relationships to put ourselves in a position that if we're so fortunate that the trials work the right way, that we can serve the community both in the U.S. and elsewhere, but that's certainly something we're focusing on. And then I think there's a significant unknown associated with the regulatory pathway itself, and we'll know better when we really start engaging, particularly since we can engage not at a theoretical level, but with actual expression with patients in hand. We're going to go to the FDA as a first step, then we'll go to other ministries of health beyond that and start chatting with them about what the right pathway is forward. We actually we have what we call Cohort C, which is a concept of a placebo-controlled trial. We need to really talk to the FDA not only about whether that's appropriate as a registration trial, but whether that's aggressive enough, frankly. And we've got to really think those issues through, and that's going to require some partnership between us and the agency. So I'd say, the regulatory pathway, and I don't, I mean that both as a challenge, but maybe in a situation where you're seeing, frankly, surprisingly robust expression and CK drops, perhaps there's also an opportunity in that to think of ways to address this issue and get therapies to patients that are waiting for them, even faster than our kind of traditional perspective might have imagined before we saw that expression level. So we need to get in front of the FDA first and foremost in the coming few months and talk to them about the art of the possible.
Yeah, I just want to add that my intention was not to question the efficacy of the results. We have been closely following Dr. Mendell, your SMA trial and the current trial. My intention was not to, question the efficacy. We see incredible results. We are very hopeful. Is
that a question?
Pat,
well,
it is
thank
notable.
you. I mean, I think that I just wanted to illustrate the potential and what we've already seen. So applying those principles to Duchenne is something I definitely would like to see, and we're doing it. So
It's just notable, this is the first time no one ever mentioned funding as top of the list for challenges. But
yeah.
My
Last question.
My question is, are there any considerations to open the studies for Becker patients or broadening the indications to include Becker patients?
I can just address the fact from GALGT2, certainly it's from that point of view, it's clearly on the list of things that we would do, anticipating both that and I mentioned other muscular dystrophies, congenital muscular dystrophies in particular. In that case where there's already primary expression of some functional dystrophin, it makes a lot of sense to consider the surrogate gene therapy, which we would expect to be at least additive and potentially synergistic. In non-human primate studies, overexpression of the AAV GALGT2 actually results in upregulation of existing dystrophin. So primates already have dystrophin. That's turned up and stabilized by GALGT2 therapy. So you would expect it would be quite beneficial to Becker patients.
Great. Well, thank you, panelists, and thank you for all your great questions.